All right, in this video, we're going to talk about cell division, the processes involved in one cell becoming two. And we're going to look at asexual reproduction and how it works in both prokaryotes and eukaryotes. So first, we're going to talk about the differences between prokaryotic and eukaryotic chromosomes. But all of them contain genetic information in the form of DNA, along with some associated proteins that allow it to form chromosomes. In prokaryotes, the cell is in a single circular chromosome, so when the cell replicates its DNA, it does it through something called rolling circle replication. It copies that one circular chromosome, and it does it pretty quickly. In eukaryotes, the DNA is in the nucleus. It's in multiple thread-like chromosomes, and the number of chromosomes varies from species to species. And so there's a different type of replication that has to happen here, but it has to happen before the cell can divide. So you can see kind of how the packaging goes. We've got naked DNA in the form of a double helix. We've got histone proteins that form a core that the DNA coils around to form a nucleosome. The nucleosomes coil up, and then they form supercoils and condenses the DNA into a chromosome, which is important because it makes it easier to move the DNA through the stages of mitosis without the DNA getting tangled up and breaking and things like that. So you got duplicated chromosome consists of two sister chromatids joined by the centromere. So in prokaryotic cell cycle, cells replicate through binary fission, which is a type of asexual reproduction. Cell copies its DNA, cell splits in two. You have two cells, each with a copy of the single chromosome. Pretty simple, pretty efficient, works pretty well for bacteria. Eukaryotes, because of the nucleus, because of the multiple chromosomes, it's a little more complicated. So they have a more complex life cycle. You can see the stages of it in the graphic here. So most of the cell's life is spent in interphase, which is the time between cell divisions. You can see interphase is the biggest part of the cell cycle graphic here. And then there's cell division, which is the time in which the nucleus divides in mitosis, and then the cell splits in two in cytokinesis. So let's talk about interphase a little bit first. Interphase is divided into three steps: G1, S, and G2. G1 are the first growth phase. This is when the cells do most of their growing. This is right after the cell is split, so it's the smallest it's going to be. The cell starts to grow. It's also making proteins and organelles that the cell is going to need to function as it gets bigger. S stands for synthesis, and it's when DNA replication, when the chromosomes get copied. And G2 is basically the period of time the cell's getting ready to go through mitosis and then cytokinesis. It makes any special organelles, any molecules it needs in order to go through cell division. This is usually the shortest part of interphase. It's not shown that way on the graphic, but that's typically the way it is. And then the M phase is when cell division occurs. So the M phase is divided into mitosis and then cytokinesis. Mitosis has four stages: prophase, metaphase, anaphase, telophase, and we use the mnemonic PMAT to remember the order: prophase, metaphase, anaphase, telophase. In prophase, the nucleus is going to condense, it's going to break down, the chromosomes are going to become visible, the spindle apparatus is going to form. If it's an animal cell, the centrioles are going to move to opposite poles, and we're going to get ready to divide. We mentioned the centromeres earlier, the place where the two sister chromatids are attached. It's also where the spindle attaches to each chromosome. The centrioles are what form the ends of the spindle at opposite ends of the cell in an animal cell. Plant cells don't have these. Metaphase, the spindle lines the chromosomes up at the center of the cell, or what we call the cell equator. You can see that here. Anaphase, the centromere splits. The spindle starts pulling the two sister chromatids to opposite poles. Telophase, the nucleus begins to reform. The chromosomes begin to go back to that chromatin state. The spindle starts to break down, and it usually leads into the splitting of the cell. Cytokinesis is the actual splitting of the cell. In an animal cell, pretty simple. The cell pinches in and the two cells split. There's a little ring that pulls and forms this cleavage furrow. In plant cells, it's a little more complicated because they got a cell wall. So there's no pinching in, but instead something called the cell plate forms in between the two new nuclei. The cell plate eventually becomes the cell wall, and now you got two cells.